will then send a process into the spinal cord and synapse directly onto the lower motor neurons that are destined to innervate the quadriceps muscle. So that when you stretch on the quadriceps muscle and set up action potentials in that primary sensory afferent, that primary sensory afferent then synapses directly onto the lower motor neuron, tells the lower motor neuron to become active, and the muscle contracts. So when you whack the patellar tendon, you stretch the quadriceps muscle, initiate action potentials in the primary sensory afferent, those action potentials then release neurotransmitter onto the lower motor neuron. The lower motor neuron fires action potentials and causes the muscle to contract. That's the sequence of events. So let's now draw that over here. So we'll draw this as the patellar tendon tap reflex. And we'll begin here with a dreadful image of a leg. And in red, this structure here is meant to be the quadriceps muscle. The quadriceps muscle has a tendon that's attached to the patella. And the patella then has another tendon which will attach it to the two bones of the lower limb, the fibula and the tibia. So then, if we look at the quadriceps muscle itself, we see that there's an input region here. There's an input region of a primary sensory afferent. If we delve deeper into this, we find that this primary sensory afferent is activated by stretch. Not really so different from the primary sensory afferent we talked about earlier at the tip of our finger, where we're beginning to get the sensation of touch. It's a very similar kind of neuron, but now its input region is embedded within the muscle itself. If we follow the neuron, we follow this neuron back, it's going to have a neuronal cell body. This is the cell body. In a region known as the dorsal, also known as the posterior root ganglion. That's the structure we have above. Now let's redraw the cross-section of the spinal cord. If we follow the process, if we were to stand on the process out here where its stretch-activated channels are located, and then follow it to the trigger zone, and then walk over the trigger zone, and then walk along this neuron, which is almost certainly going to be heavily myelinated, walk all the way into the dorsal root ganglion, climb over the cell body, climb over to the other side, get on the other extension of this neuron, we'll now follow this into the central nervous system. So again, this is the dorsal root ganglion, this is the dorsal root, and the neuron process is entering the structure called the dorsal horn. That primary sensory afferent will extend all the way into this region down here, known as the ventral or anterior horn. In the anterior horn, that primary sensory afferent now synapses onto a lower motor neuron. This neuron now sends a process out into the periphery, just as we saw above. It enters this structure here in the periphery, known as the ventral or anterior root. The neuron enters the spinal nerve and goes all the way back here to the quadriceps muscle. And what I'm trying to convey here is this lower motor neuron synapses. The lower motor neuron synapses onto the quadriceps muscle. So now let's follow the pathway that I showed you by sitting on the table. Come out and you whack the tendon here. The patellar tendon gets stretched and pulled. That in turn, because of the connection of the patella to the quadriceps muscle, pulls or stretches the quadriceps muscle. Because the stretch receptor neuron, the primary sensory afferent, is embedded in the muscle itself, we're now going to stretch the input region of the primary sensory afferent. We stretch it enough, we open sufficient numbers of stretch-activated channels. They provide sufficient positive charge in the form of a generator potential that summates at the trigger zone to fire action potentials all the way into the central nervous system. We can think of it as a single action potential if you'd like, but more likely it's going to be a whole bevy of them, and it will be a whole bout of action potentials, maybe 10 or 100. Then when those action potentials arrive here at the output region of the primary sensory afferent, they're going to release a neurotransmitter, almost certainly glutamate, that now will be released by the story we talked about the other day using the influx of calcium and the snare proteins. They'll release neurotransmitter onto the lower motor neuron. They will release so much neurotransmitter here from many, many sites that they almost invariably will provide enough excitatory postsynaptic potentials to bring this membrane to threshold to the trigger zone. That then fires action potentials to propagate into the periphery down to the muscle and tell the muscle to contract. So now you have the circuit. This is the diagrammatic representation of the circuit that I just described, but when you sit on the gurney at the doctor's office. So let's look at this for a moment and look at the physiologic and the anatomic structures that we're dealing with. The potentials that are generated at each step of the way. So out here we're trying to represent the input region of the primary sensory afferent within the muscle. This little roundabout piece here is meant to be the input region of the primary sensory afferent where we have aggregated the stretch-activated channels. Right here, perhaps, there'll then be the voltage-gated channels that are now triggered to open by the opening of these stretch-activated channels. These then will summate, add together, and if they're large enough, they'll generate an action potential which now propagates along the primary sensory afferent, over the neuronal cell body and dorsal root ganglion, and into the central nervous system. And in the central nervous system, this primary sensory afferent now will release neurotransmitter by the mechanisms we talked about earlier this week. That neurotransmitter released from the primary sensory afferent now binds to receptors, glutamate receptors, almost certainly here on the lower motor neuron. Those ligand-gated channels open, they generate an excitatory postsynaptic potential shown here. And in this case, almost always we have so many of these excitatory postsynaptic potentials, they will summate at the trigger zone. Action potential now that propagates all the way down to the output region, releasing the neurotransmitter here, acetylcholine, that binds to ligand-gated inotropic channels, opening them, leading to action potentials in the muscle fibers, which cause the muscle to contract. So that's the circuit. That's the series of electrical events that occurs in the circuit. Now, this shows the circuit that we just described. And we're going to add one more piece here in this figure. So again, let's walk through it. Anatomically, out here in the quadriceps muscle, we have the sensory input region. This is where the stretch receptor channels are, stretch activated channels. So we initiate an action potential, propagates into the central nervous system, and the circuit we followed was this blue neuron synapsing onto the green lower motor neuron, which then immediately goes out and synapses onto muscle, the quadriceps muscle. So that when we stretch the quadriceps muscle through this circuit, we get the muscle to contract. Now, there's one other piece that I haven't put in here yet that I want to emphasize here. You'll notice there's a branch of this primary sensory afferent. That the branch of this primary sensory afferent synapses onto this red neuron here. That red neuron is confined to the gray matter of the spinal cord, never leaves. It's always there. It's always in between. It's said to be an interneuron. It's an interneuron between the primary sensory afferent here and this lower motor neuron. In this particular case, that interneuron is an inhibitory interneuron. So watch what happens here. We activate action potentials at the input region here in the primary sensory afferent. We followed the release of excitatory neurotransmitter onto the lower motor neuron. We've done that circuit. But the same thing happens at this interface. That is, this primary sensory afferent is synapsing onto the interneuron. The primary sensory afferent is releasing glutamate. That's turning on the glutamate receptors here in this interneuron. If we get enough glutamate release that we activate enough synapses that we generate an action potential, then we have an action potential in this interneuron. The action potential arrives at the output region. The process we talked about yesterday is then followed, and we, through the snare hypothesis, release neurotransmitter. But now the neurotransmitter releasing is GABA, or since we're in the spinal cord, probably glycine. But regardless, it's an inhibitory neurotransmitter. So then what happens is by releasing the inhibitory neurotransmitter, we are inhibiting this lower motor neuron. So now one of the issues that comes up here is that you begin to think that, well, this one's on and this one's off. But remember, that's not exactly the way inhibition works. Inhibition is a kind of balancing act, such that if you now have more inhibition onto this lower motor neuron, it's less likely to fire action potentials. And note where this lower motor neuron innervates, where it has its output region. This magenta lower motor neuron goes out and innervates this muscle here, which is the hamstring muscle. That's the antagonistic muscle. And I'll put this on the board in a moment, but recognize that all of our muscles are laid out in antagonistic pairs. We'll come to this on Monday. Our muscles are laid out in antagonistic pairs so that when this muscle, the quadriceps, contracts and it shortens, it's going to work over the knee joint and kick the foot up. But then when we want to bring the foot back into its original position, we now contract the hamstring muscle and bring the foot back to its original position. And in the process, then, we'll re-stretch the quadriceps muscle and have it reset. The muscles cannot reset on their own. They have to have some energy put into them to re-stretch them to their original position. But now, again, here is the site of inhibition. So let's have a look on our drawing. So the process we followed here was for the quadriceps muscle, but the antagonistic muscle down here in purple is now the hamstring. Now, if you wanted to flex this joint, if you wanted to take your foot and bring it up to your hip, then what you would do is you would contract the hamstrings muscle and relax quadriceps muscle. But in our tendon tap reflex, we have contracted the quadriceps. And what we wanted to do was simultaneously then turn off the hamstring. So here's a branch of the primary sensory afferent. It synapses onto this green interneuron. The green interneuron is inhibitory. And now this orange neuron, this orange lower motor neuron, is now going out and it's going to innervate the hamstrings muscle. But recognize out here that this is a synapse between the lower motor neuron and the muscle. That the neurotransmitter here is going to be acetylcholine. It's just an excitatory neurotransmitter that's going to tell the muscle to contract. But the way we turn this off is in the central nervous system. We inhibit this lower motor neuron to the quadriceps by simultaneously activating the lower motor neuron. We inhibit this lower motor neuron to the hamstrings by simultaneously activating the quadriceps, but at the same time activating the inhibitory interneuron to turn off the motor neuron going to the hamstrings. So these guys are working in antagonistic pairs. When we turn one on, we turn the other off. But the integration pattern out here, the innervation pattern out here of the lower motor neurons is exactly the same. That is, we have a lower motor neuron to muscle, releasing acetylcholine and being excitatory. The only place where we inhibit these guys is in the central nervous system. So any questions to there? So one of the issues, this is perfectly fine, it's perfectly true. You could follow this through from the other side. But one of the surprising findings is that if we cut the spinal cord here, so that we break the spinal cord, we break connections between the spinal cord and the brain. If we break those connections, this remains entirely intact. It's a local reflex. So this local reflex occurs simply because of the innervation of the primary sensory afferent onto the lower motor neuron that then goes out to the quadriceps muscle. Now we could do this with any number of muscles all over the body. We have these circuits. But what I want to emphasize here simply is that this goes to the quadriceps muscle. This remains perfectly intact, but recognize we now, by cutting the cord, we have lost the connection between the spinal cord and the brain. So this raises the question of how do we manage to get information from the periphery up to the brain. In the last minute, let me draw this in, and then we'll come back to this tomorrow. This primary sensory afferent comes into the cord, and not only does it enter the cord, it also sends a process all the way up to the brainstem. This process here is going to be located in the white matter here called the dorsal columns. So this neuron, which we show in the longitudinal section, now comes into the cord, sends a branch, and enters the dorsal columns. Now just let me make one more synapse here and let you go. Up here in a region known as the dorsal column nucleus. Remember, nucleus is where every input regions. That primary sensory afferent synapses here onto a second order neuron. And that neuron will cross the midline, and we'll pick this up tomorrow and follow this pathway all the way up to the somatosensory cortex.
Is doing two things. It's communicating to the lower motor neuron and to the...
Yes, and it's also communicating to the inhibitory neuron.
Okay. Is the dorsal column another neuron, or that's just another...
Whenever you see the dorsal column is a tract. It's a white matter tract. And so it's now going to be carrying information up the cord, and we'll pick that up tomorrow.
Okay. That's just happening at the same time as the primary sensory afferent communicating with the lower and the inhibitory.
That's right.
Okay.
How's it going?
Good, how are you?
All right.
Primary sensory, the only ones that have stretch activated channels, or just for this contact?
Well, what?
Our sensory afferent neurons, are those the only ones that have stretch activated channels, or?
No. There will be different primary sensory afferents. Some will be for pain, some will be for temperature, some will be for pressure. And some will be for fine touch, some will be for proprioception. And we'll talk about that tomorrow.
So there's just one that's just for stretching.
Right, there's a subset. There'll be a whole series of them for stretch. Some of them will be embedded in muscle, some of them will be in your skin.
Okay.
All right?
So I don't think you think this is super important, but I wanted to ask you real quick just to make sure. It was on that other drawing, but I think she got rid of it. So the dorsal root, the dorsal horn, and the dorsal root ganglia, those are three different places on that.
That's right. There are three different
Locations.
structures. We give them structural names, but they're really different way stations.
Okay.
So when you see ganglion, that's neuronal cell bodies.
Okay.
When you see root, that's just, it's spelled R-O-T, but you can think about it as R-U-T-E into the central nervous system.
Yeah.
And then once we get into the central nervous system, that's now going to be the horn.
Okay.
And the horn is gray matter. And that's where synapses occur.
Okay. Okay. And so when you have after the horn, or it was not the horn, it was a small little structure. You drew a circle right here, and I forgot what it was called. What was it?
Oh, no, I didn't draw it here. I drew it all the way up in the brainstem.
I drew it further out. Oh no, I'm trying to figure out. I must have been talking about the horn, but you said immediately once you get out of that, then it becomes the peripheral nervous system.
Oh, okay. That dotted line's drawn here.
Okay. The peripheral nervous system is immediately after you get out
Right,
of that. Not
right.
After, okay.
And equivalently down here, there's on the ventral side, the anterior side, this will also be in the PNS.
Okay.
So anytime you get outside this magical boundary. So remember, and the only reason you'd have to hang your hat on this is if we get central, those would be all like endocytes. If we get peripheral, it'd be Schwann.
Okay, appreciate it. Thank you.
Sure.
So I'm in Gamma Beta here, and I just want to stand here to show that I was in class today because I had to delete and repeat the course, and they're like worried about it.
All right.
Super simple, I know.
Okay.
Nervous system and all of their cell bodies each housed in there. It's like it's where all it's what we call the congregate of them all. Right, on one neuron it's a cell body, but when you have all of these neurons in the peripheral nervous system, they travel together in a nerve.
Oh, you lost an eraser?
Nerve and neuron.
Well, I do care to some extent because I tend to put them on the sideboard so when I have
An axon can travel in nerves, the neurons, and then when the
somebody else lost another.
we call that a ganglia. And then when the cell bodies go along in the central.
And then,
No. We call it a nucleus. It's just a nucleus.
oh well,
Yeah.
we'll manage.
So the horn is just a region of the spinal cord. It's that butterfly kind of thing. Yeah. Okay. Cool. Yeah, no worries. Thanks. Did she tell you that everything crashed on us while we were filming that?
No, that's why she was a little distant. Okay.
Yeah.
Oh no.
Yeah.
We lost it.
Yeah. It was a mess. The camera glitched and like,
Did you get the blue screen of death?
yep.
Oh shoot, we've got to get that fixed.
Got the blue screen of death.
Clear the cache.
I was so proud of myself. I had it all nice. I had everything set up right.
How far did you get in?
Probably 20 minutes.
That seems to be when we get this glitch.
Just like, was like, well, nope. Sorry. So you want me to just clear the memory card and start over?
No, don't clear the memory card. Clear the cache on your computer.
How do I do that?
I don't know.
It reset and I followed the instructions on.
Okay, Andy will tell you what to do.
But Andy's in an interview.
I know. Well, I think there's the memory is cached on that, and I think what happens is we fill the cache and then it freaks out.
Kendra's like, I thought we had fixed it. Apparently Andy had done something that
Right.
had fixed it.
We thought we had fixed it, but apparently not.
So we got the blue screen of death. We got a mess. And then the camera glitched once and stopped recording.
Why is that?
I don't know. It wouldn't like one of the settings numbers. It wouldn't change. Like I would like the focus changed, but the number itself, and it seemed like when the whole thing caught up, it just kind of stopped recording.
Okay.
So it was
Well,
nice.
let's hope the
So
second one is better than the
wasn't
first.
Great. It was awesome lesson.
All right.
But I told her, I was like, of course.
Well, you know, we can always mount the ones from last semester. They're a little off,
I was
but
like, the first time I do this, and I'm like, it's all nice and set up.
well, we had another blue screen of death earlier this week. I'm gonna have to call Terry,
Okay.
as we're having this is too frequent.